The next lecture is title is Understanding Transformation between Decision Potent and Decision-null Representation in Visual Working Memory using demixed PCA, presented by Quan, PhD student in the Dr. Postel lab at University of Wisconsin, and I think that we will learn too much here. Let's start by Quan, and just please share your screen and start. We have it.
Can you hear me?
Yes, hear you.
And see my screen?
Yes, just put the full, yeah, it's great.
Okay, cool. Alrighty. So good morning, everyone. In our daily life, we constantly need to juggle multiple pieces of information held in mind. For example, at a cocktail party, you're introduced to Bob and Jim, and you hold these names in your working memory. When you go up to talk to Bob, his name is prioritized in memory. Because you want to talk to Jim later, you still hold on to the name Jim, but at this time, it is unprioritized in memory as you talk to Bob. And later, when you go talk to Jim, the opposite happens. To study the bases of attentional prioritization, we use an age-old working memory task called 2-back. This task requires subjects to constantly compare the current stimulus with the one that was presented 2 times ago. Let's focus on item N here for an example. Say this is when you first learned the name Jim, and then you wanted to talk to Bob next. So during this time period, the name Jim is an unprioritized memory item, or UMI. And after that, when you want to talk to Jim next, UMI. And you want to talk to Jim next, his name becomes a prioritized memory item, or PMI. To study the neural representation of the stimulus as a function of behavioral priority, in previous work in our lab, we recorded EEG during this task and showed that UMI and PMI are represented in the same code or subspace, but in different and opposite formats. To understand how the representation is transformed between UMI and PMI, we trained a recurrent neural network with LSTM architecture to do this task. Briefly, stimulus information in the form of a one-hot vector goes into these 6 stimulus selective input units. Information is then propagated into this LSTM layer, which in turn projects to the output unit and yields the decision, match versus non-match. To look at the internal representation of the network, we did a PCA on the activity patterns in the LSTM layer and projected them onto the first 2 PC dimensions and mapped them back onto this 2-back time course, which we call a trial. The network is run on many trials. Here, each dot is a stimulus representation within a single trial at this time point, and the color always indicates N, or Jim's, identity. And we track N's representational structure of all the trials over time. After stimulus N is presented, its representation falls onto these roughly parallel color planes, and the structure rotates roughly clockwise as N transitions from UMI to PMI to finally compare with N+2 to make a decision. In this talk, we mainly focus on these 2 delay periods. To characterize the rotational transformation from UMI to PMI, we used a dimensionality reduction method called demixed PCA. Unlike PCA, dPCA ignores trial-to-trial fluctuations and can tease out sources of variability only dependent upon the stimulus condition, which we care about. It allows us to find UMI and PMI specific subspaces indicated by these black arrows. Some results. Here are the dPCA projections from an example neural network. The axes for each figure are the top 2 dPC components, and the first row is the UMI subspace, and the second row is the PMI subspace. The columns are stimulus response averages based on UMI versus PMI. So in each subspace, notice that the representational structure shown by the order of the colored dots from left to right is very similar between UMI and PMI. Actually, if you rotate the UMI representation clockwise, it will match the PMI data and vice versa. The fact that we get roughly the same representation in each column suggests that UMI and PMI are coded in similar dimensions. Here are EEG data for an example subject. Again, stimulus structures are similar column-wise, meaning UMI and PMI are coded in similar subspaces, consistent with results from our previous work. And row-wise, if I rotate the UMI figures on the left, they can be aligned reasonably well with the PMI data on the right. Just to orient you, for example, in the first row, if we anchor this figure with red, blue, and cyan, we can see red, blue, cyan on the right figure, and the yellow and purple are in the middle, and the green is aligned less well. And on the bottom row, if we align the figure with green, cyan, blue, green, cyan, blue, and again, the yellow and purple are in the middle, and the red is aligned less well. We can see that the alignment of the EEG isn't as good as with the RNN. We think it is because EEG data is very noisy and high dimensional, and the rotation may happen in other dimensions as well. It's also possible that representations of orientation stimuli in the brain are non-linear, so they can't be well captured by linear compression of the data. It is still a work in progress to get a bit clearer picture on this, and overall, dPCA is tailored to extract signals of interest and should allow us to qualitatively and in the future quantitatively characterize and compare the RNN and EEG representations. I'd like to thank my lab and funding and also my awesome collaborator, Jorge. And here are some next steps and challenges. And I'm happy to take any questions, comments, and suggestions and clarify anything that was unclear. Thank you.
Thank you, Quan, for your talk. And is there any question by anyone? Can you send your question in the question button and we will ask. No question?
It's probably too early for people.
Okay. There's some question. Jack Stewart asked, I think that is normally the case that the UMI item are harder to decode in PFC, for example, than PMI item. Does the LSTM network display this as well?
Not really. So in the LSTM network, the decodability of UMI and PMI are similar. Yeah, again, it is not the brain, and it probably doesn't have the brain-like representational characteristics. Yeah.
Hadi Motamedi has a question. Hadi, you can unmute your microphone and ask the question yourself. You is panelist.
Okay. Great. Hello, thanks for your great talk. It was really awesome. My question is that from EEG data, it's very cool that what you're saying is we can know which working memory it's capturing, right? It's visual task, if I'm understanding it correctly.
Yes.
Yeah. So in this visual task, you can decode from the EEG which working memory or which image actually the participant is seeing and how long, for example, it's keeping it into this memory, something like that.
Right. So you can imagine in the two-back task, after the presentation of each item, say the item is item N, okay? So after presentation of N is unprioritized, whereas N minus 1 is prioritized, right? So you basically, it's the same neural data, but you can decode it using or reconstruct it using the PMI label, which is N minus 1, or the UMI label, which is N. So that basically can dissociate which working memory it is.
Okay.
Yeah. Okay, thank you.
Thank you, Adi. And another question by Dr. Paulson. Forouzan, can you bring him on, ask his question?
Oh, yeah, okay. So Brad asks, your RNN only has one hidden layer. How might you describe? Sorry?
There is Brad is on.
Oh, okay.
Yeah, I was just following up on the question from Jake Stroud, right? I think that's what he's asking. And so the next step, I guess, would be, how would you represent different brain areas? Presumably, you'd need different layers in your RNN, right?
Right. So I would say, so right now it's just LSTM, and it only has one layer, and it doesn't have the distinction of PFC or parietal lobe or occipital lobe. But we can imagine we can possibly extend the network to a more biologically like a realistic network. For example, the well-established PBWM, the prefrontal basal ganglia striatum network. So we can model some features or cell types and of different brain areas and connect them with connections that are constrained by empirical data. And that way we can better tease apart what kind of representation or where the representation lives.
Okay.
Yeah, so one comment, just to continue the discussion on this, one comment I'd make is that right now like Shwan was just saying, the LSTM is just one homogeneous network, and from the perspective of the task that it's trained on, both the PMI and the UMI are equally important. So clearly it's going to be representing both of them equally faithfully. But if we start to add some more structure into that LSTM, where maybe some areas are playing more of a role in doing PMI or less of a role in doing UMI, et cetera, then maybe some of these differences might arise that Jake pointed to. But yeah, I think it's an interesting question.
Okay, and another point that Sanchit Gupta is asked, I might have missed it as I joined halfway. I understand, but she said, can you unmute microphone and ask your question?
Yeah. So I want to ask, like, how exactly do you come up with that dPCA subspace? As I missed your first part, so if you can briefly tell, that would be great. Thanks.
Right, sure. I can show you the dPCA algorithm. Let me just share my screen real quickly. So can you see it now?
Yes, obviously.
So this is the essential dPCA algorithm. So what it does, let me parse it out for you. So here the X bar is the grand mean of the data, overall trials and time points and stimulus conditions. And the XS is the mean response for the stimulus S. So this term captures how the stimulus means fluctuates around the grand mean, or the stimulus specific variance. And here we have the response on time point or trial I for the stimulus S. And this basically shows how each trial or time point fluctuates at the grand mean. And what the dPCA does is that it has a decoder vector and an encoder vector. And the goal of dPCA is basically reduce this reconstruction error, which is the expectation over all stimuli and time points. And this way you can basically capture the variance that's only specific to a certain like task parameter. In our case, it's the stimulus. So basically, we use this W vector. Well, you can imagine like PCA, you can order the different components by the variance explained. So we can choose the top two dPCA components to construct the two-dimensional subspace.
Okay, thank you. And what is the size of X? Is it the number of electrodes in our EEG?
The number of electrodes, so we did two analyses, right? We did it with RNN and also EEG. So with EEG, yes, the number of electrodes is 60.
Okay, thank you.
Yeah.
Okay, thank you. And any other question that